Hello everyone. Today we are talking about a new topic. This is magnetic resonance molecular imaging. I'm Yu Dexin. I'm a radiologist in Qilu Hospital of Shandong University. At first, we are talking about the concept of this topic. Now, let's begin the first part of our presentation of the concept of the magnetic resonance molecular imaging. This imaging is a science that uses imaging method to display specific molecules at the tissue level, cell, and subcellular levels, reflect changes in molecular levels in living states, and quantitatively study to biological behavior of images. MRI molecular imaging is an important branch and component of the molecular imaging. MRI is used to image specific biomolecule in view for the purpose of the early diagnosis of the lesion, specific diagnosis, and effective monitoring. Let's talk about it in the following parts. The first is the development of the magnetic resonance imaging. Then we'll talk about methods of the implementation. Then we'll talk about the methods we have used, and then we are talking about the imaging process. Finally, we'll give you some examples. In recent years, the rise of the MRI molecular imaging has greatly promoted the development of the medical imaging, making medical imaging from traditional imaging from anatomy and pathology to the changes at the molecular and cellular levels. Broad molecular magnetic resonance imaging also induces functional imaging. Functional MRI is a research method that causes nerve activity in the similar part of the brain cortex by stimulating specific sense and display it through MRI images. The following three images show conventional MRI, anatomical imaging, molecular target imaging, and functional brain imaging. At the cellular level, the cells labeled with molecular imaging in vivo have been successfully used to monitor the infiltration of inflammatory cell in lesion and the migration and differentiation of transplant stem cells in vivo in cell transplantation therapy. At the molecular level, the occurrence and development of the disease can be dynamically observed by markers that can specifically recognize and bind to target tissue. Multiple biological events can be monitored at the same time and studied in time and space. The processes include abnormal cell metabolism, abnormal expression of the receptors on cell surface, abnormal enzyme activity, and apoptosis and genesis of the tumor. At gene level, image of the report gene can indirectly reflect the expression of the target gene, and successful realization of pairing of the target genes has been achieved. In vivo monitoring of gene therapy and its application in this study of the tumor genesis, growth, metastasis, and other characteristics. So, we are talking about the method of the implementation. There are three requirements for the MRI molecular imaging, including a specific marker, MRI equipment, and magnetic molecular probes for the lesion. This brings up one of the key to molecular imaging, which is to have sensitive MRI imaging equipment. In the detection of the trace substance, MRI is only at the level of micromole. Therefore, high-resolution MRI imaging equipment is one of the key to drive the molecular imaging process. At first, we should understand the pathway and the mechanism of action of the cells and molecules. We want to image what will pave the way for us to find suitable targets. Then, we conducted a series of cell-level experiments and verification on the carrier we used, including the biosafety of the carrier and its image effects. What we need to do this to first verify and test in VM at the animal level, such as imaging effects, therapeutic effects, and so on. Finally, we can enter next preclinical experiments to provide some hint for the future clinical research and diagnosis and treatment. Finally, we can enter next preclinical experiments to provide some hint for the future clinical research and diagnosis and treatment. The image on the left shows an MRI device for small animals, and this image on the right shows a series of hardware devices for the device. Let's look at a few examples of the molecular imaging. This image on the left shows a high-resolution image of the living brain and the spinal cord structure. This image on the top right shows a 3D image of the tiny intracranial blood vessels, and this image on the bottom right shows a high-resolution molecular diffusion weighted image. Okay, that's all for this part. Thank you very much.